So welcome, my dear friends. So today's our resource person, young and dynamic personality, my beloved brother, Dr. Gogikar Bhageerath Garu, Assistant Professor, Department of Botany, Government Degree College, Rangasaipet, Warangal District. Sir has huge experience in the field of plant sciences, especially plant molecular biology, genetics. So, Sir genetic expert. Sir is uh, since last since 2022. Sir has given his services, valuable services for our young aspirants. Sir, due to his services, most of the young budding botanists, they are get benefited and join in various institutions, university affiliated colleges, CPGET 2022, 23 and 24. So in this regard, CPGET 2025 also, SAR is ready to going to serve to present his most expertise and most important topic in plant biology and plant uh, genetics, especially genetics. In this regard, Dr. Gogikar Bhageerath Garu, Assistant Professor, Head Department of Botany, Government Degree College, Rangasaipet, Warangal. Sir, please welcome and share your screen. Start your session, sir. Please welcome.
Thank you, sir, for giving this opportunity. And I think screen is visible to everybody.
No, sir. No.
No. Once I will. Now?
Yes. Now screen is screening, but not yet visible.
It visible?
No, sir. Now it's sharing. Yes. Yes, sir. It's visible. Please, you may proceed.
Okay. So first of all, thank you to Dr. Padala Tirupati, CPGET 2025 convener. Since 5 years, sir was taken initiation for this program, and most of the students are benefited through this online CPGET PG entrance coaching. It is really a wonderful program to especially public funded institution students, because they can't affordable in all aspects. That's why it is a free learning process, but even though we are getting more knowledge and current knowledge, up-to-date knowledge regarding in our subject, that is botany. So hence I congratulate and as well as thanks to Padala Tirupati for giving this opportunity. So today we will discuss about plant genetics. It is a branch of biology and very important with respect to molecular biology and other aspects. So today our class is mainly focused on introduction of the genetics, origin and etymology, history of genetics, and what are the timeline of research in the genetics area, how it is useful in the elucidate different type of modern information regarding the genes or the chromosomes or the DNA, and how these genetics branch is divided into subdisciplinary branches, and what is the importance of the genetics. All these things we can learn in this class as a small, shorter way. Okay, so next slide. So first of all, in every organism, every cell hold a secret script. In every cell in your body, it holds a secret script. That is nothing but it is a genetic code that decides who you are, how you look, and how your body functions, and when even how might to respond to a drug or a disease. That is indicates what that overall summary is indicates the one of the important branch, that is genetics. Genetics is a branch of biology which dealt with heredity and also genes as well as variations. It is a central pillar of the biology and also which links to other areas like especially agriculture, medicine, and biotechnology. So genes are, we can say simply, genes are coined as units of heredity. Units of heredity. Genes are made up of tiny parts of the DNA, that is deoxyribonucleic acid, you know very well. So these are all things already learned. First, we can go for how this genetics branch evolved. So during the 19th century, we know, laid down a stone for the genetics branch by a famous person, that is Gregor Johann Mendel. You know very well, Mendel is done experiments, hybridization experiments in pea plant, and his work later provided with the mathematical ratio information of the science of genetics. In this 19th century, the genetics branch is originated. That's why the Gregor Johann Mendel was coined as father of classical genetics. Why he is popular as a father of genetics? Because first time he discovered the inheritance laws. These are the three things you learned in the lower class also. Inheritance laws, first time discovered by the Gregor Johann Mendel. Before 19th century, we don't know about the genetics. We don't know about the how the parental characters are transmitted to children. My voice is clear? Everyone?
Yes, sir.
Okay.
Yes, sir. Your voice.
Okay,
Yeah.
okay. Very nice. So the genetics, we can broadly divide it into two category, two areas. One is classical genetics and another one is modern genetics or molecular genetics. So before going to the classical genetics, how the mainly agriculturists are focused on selective breeding of crops and animals. So earlier agriculturists are know the causes of variation was hidden in sexual reproduction. That means a excellent sperm combined with each other and it leads to formation of zygote. Zygote later developed as embryo. Embryo is given a birth to new infant or new organism. So the agriculturists and when dawn of civilization is occurs in the course of evolution, man focused on the how the variation is comes. So he don't know, but what is the mechanism of variation? But he observed which is hidden in the sexual reproduction only. That matter is identified in the earliest era, that is 8,000 to 10,000 BC. Because of it, they could successfully blend domesticated varieties. That's why man is focused on domestication of animals and plants through the selective crossing and artificial selection of, in example, Indian cow, Shival of Punjab, and a domesticated form of an ancestral wild cow. So our ancestors had very little idea about the scientific basis of inheritance and variation. అంటే మన పూర్వీకులు చాలా తక్కువ సమాచారాన్ని కలిగి ఉన్నారు. దేనికి సంబంధించి? ఏ రకంగా ఇన్హెరిటెన్స్ అనేది ఏ విధంగా జరుగుతుంది, వేరియేషన్స్ వైవిధ్యాలు ఎట్లా వస్తాయి అన్న దాని మీద వాళ్ళకు చాలా చాలా లీస్ట్ ఇన్ఫర్మేషన్ ఉందని చెప్పచ్చు. మరి దీన్ని ఎలా రివీల్ చేయగలిగాడు మెండల్ అనేది మనం క్లాసికల్ జెనెటిక్స్ ద్వారా చూడొచ్చు. సో హౌ ఇట్ ఈస్ జస్ట్ యూ కెన్ ఓకే. Okay. So genetics branch is broadly categorized or divided into two. One is classical genetics. Classical genetics is mainly focused on the inheritance of traits based on observable outcomes. Whatever the morphological features observed in any plant or animal, based on the characteristics only, how the parental characters are transmitted to the children or offsprings. That is the main focus observed in the classical genetics. But once comes to the modern genetics, it is completely different. How it is differ in these aspects? First of all, first we can go for time period. The classical genetics is revealed in mid-19th century to 20th century. So modern genetics is evolved mid 20th century to present. Why in the mid 20th century we are getting more information about the genes? Why? Because our science and technology gradually develops. So many instruments are, so many principal scientific methods are established in the research programs. That's why we know about the lot of information about the genes and DNA and replication, translation, transcription, such type of physiological process are done in the cells. This information is revealed about the genes also. That's why the modern genetics is evolved in the mid 20th century to present genome editing also. It comes under modern genetics. So who are the founders? Gregor Johann Mendel is the founder of classical genetics. Different various scientists are contribute their services in the modern genetics. Example, Watson and Crick, Franklin, Biddle, Tatum. So many scientists are there. So the main focus of the classical genetics is transmission of traits from parent to offsprings only. But in modern genetics, we learn about the structure of the gene, function of the gene, and how the genes are regulated at molecular level. How a particular phenotypic character is established in the plant, how the chromosome, how the gene is revealed, how the gene is respond, and at molecular level, how how it regulate, at what time the phenotypic feature is outcome. These are all things are molecular level we can understand. It is very important in the modern genetics. Unit of study here, gene as an abstract and invisible factor. According to the classical genetics, gene, we can't know about the gene. Asal gene ante ఏంటో తెలియదు. That's why Mendel says the gene is called as factor or element. Gene అనే పదాన్ని ఫస్ట్ టైం ఎవరు గమనించారు? జోహెన్సన్ అనేవాడు ఫస్ట్ టైం కాయిన్ చేశాడు జీన్ అనే పదాన్ని. జెనెటిక్స్ అనే పదాన్ని బేట్సన్ అనే సైంటిస్ట్ ఫస్ట్ టైం డిజైన్ చేశాడు. అసలు మెండల్కి జీన్ అంటే తెలియదు, జెనెటిక్స్ అంటే తెలియదు. బట్ హీ కాన్సన్ట్రేటెడ్ ఆన్ హౌ ద పేరెంటల్ క్యారెక్టర్స్ ఆర్ ట్రాన్స్మిట్ టు హాఫ్ స్ప్రింగ్స్. వాట్ ఈస్ ద సీక్రసీ ఇన్సైడ్ దట్ హీ ఈస్ ఎస్టాబ్లిష్డ్ బై done the hybridization experiment, and the results are framed in a mathematical ratios. Based on the mathematical ratios, he discovered the inheritance laws. That's all. So here, according to the classical genetics, gene is as an abstract or invisible factor. But modern genetics says gene is nothing but a smallest part of the DNA. That is nothing but one cistron is also we can call as gene. It is a functionable cistron. It exact copy, it giving a result product of a protein or enzyme or a sensor. It act like a molecular sensor. So the gene as a segment of DNA according to the modern genetics. What are the tools generally used for the outcome of the results? So in the classical genetics, they are using only crossbreeding, phenotypic ratios, Punnett squares only. But modern genetics, we are using different type of scientific things, cellular techniques, DNA sequencing, PCR, gene editing tools, CRISPR-Cas9 technology. So RNA editing, RNase technology. So different type of technology and cellular techniques, principal scientific methods also we are used. Experimental organism, pea plants and Drosophila here. Here so many bacteria, virus, mice, humans, all are used as experimental tools. Key concept is here dominance, segregation, only independent assortment. But here gene expression, what is the genetic code, how mutations are occurs, what is meant by epigenetics, what is meant by genomics, proteomics, like that. Here data type is only phenotypic traits. We can exactly observe the only traits, only characters only here. But here we are using DNA sequences, proteins, gene expression profiles, and we are applications only in plant animal breeding. But modern genetic applications are we are using in different sectors, medicinal sector, biotechnology sector, gene therapy, genomics, and the limitations are there. Some explains how the gene works are there. You can see this. So we can go for the next slide. Once. So what is the classical genetics is laid the foundation using observable traits and statistical ratios only. What about modern genetics? Modern genetics explain the molecular basis of heredity using DNA and advanced technology. So molecular genetics only focus on the structure and function of genes at the DNA level, studies gene replication, transcription, translation, mutations, and we are using number of tools like PCR, gene cloning, CRISPR, Cas9 technology. How these genetics divided into subdisciplines. So biochemical genetics is there. So that indicates, it denotes that chemical nature and metabolic role of genes. Explain how gene mutations cause metabolic disorders. In your zoology subject, you studied alkaptonuria, phenylketonuria. These are all disorders are there due to the metabolism, metabolic disorders. So due to the failure of the gene, how the gene mutation causes metabolic disorders. So it is basics of inborn errors of metabolism. You know very well, Gerard is the father of experimental genetics, according to the biochemical genetics. Next one is population genetics. You know very well, studied in botany and zoology and microbiology also. Studies is gene frequency and gene variation. How the gene pool, a population containing some of the genes, the gene pool changes its gene frequency due to genetic variations within the population. So it deals with evolution, natural selection, and genetic drift. These are the three words we are studied in the evolution chapter. It is foundation for evolutionary biology and conservation of the genetics also. You know very well, in the population genetics is one of the important area, that is Hardy-Weinberg law. You definitely got one problem on Hardy-Weinberg law on gene frequency, or the dominant gene frequency, or the recessive gene frequency, like that. Next one is cytogenetics. It is related to only cytology and genetics. It is a combines when cytology and genetics. That means nothing but cytology. We are studied about the nucleus,